Hello everyone. Hope you are doing well and are safe. Welcome back to Weekend Chat with Researchers Season 2, and this is our final session of this season. As you all know, we are Bugbears group at NCBS, which is headed by Dr. Ashwin, and this is our science and research outreach channel. Through these sessions, we aim to bring research closer to science enthusiasts, and we believe anybody sitting at home can understand science and research irrespective of one's background. And I am very happy to announce that yesterday only it was a anniversary, a 1 year anniversary to start these outreach sessions. And we thank you for being with us in this journey. So with you all, I am Nitish, and with me, Akshara and Atal will be the host for this session. Hello, Akshara and Atal.
Hi.
Hi, Nitish.
Great. So we will proceed with the session, and we will. I'll just ask Akshara about how about you tell our viewers what our session is going to be.
Yeah. Hello, everyone. So, in our past session, we have discussed many aspects of bacterial biology. While in continuation to that, today in our last session of this season, our guest will explain us about different aspects of bacterial cell cycle, how they regulated, what are the internal and external cues.
Oh, great. So another microbiology session of our season, and we hope you stay us throughout. And there's some surprise which will be coming to you guys. So Atal, since you are the newest member of our group, how about you tell our viewers who our guest is?
Sure, Nitish. Hi, everyone. Today we have with us Dr. Sunish Radhakrishnan, who is an associate professor at ICER Pune. Sunish's group is broadly interested in understanding prokaryotic cell biology and development. Sunish has been tracing the mysteries of cell cycle control, differentiation, and developmental signaling in bacteria, and that is from quite some time. In the past, he has also done signaling in bacteria and has done some research outreach sessions for high school students. Also, this year he has been honored with a senior fellowship by DBT Wellcome Trust Alliance. Welcome, Sunish. We are glad to have you here with us in the final session of season 2.
Hello, Sunish.
Hi.
Hello, welcome to
Thank
our
you.
group. Great. So, without delaying much, we'll kickstart this session, and I'll just ask very simple question. When I saw posters around about what is actually a cell cycle. I mean, this cell it does something. So, what actually is cell cycle?
So basically, when a cell is there, the primary aim of the cell is to divide and proliferate itself, isn't it? So, and what consists of cell cycle is various events that leads the cell to grow, helps the cell to grow, replicate itself, that is, divide itself, and then proliferate. So, the different events that controls this growth, division, and proliferation is classified into various stages in cell cycle. For example, I can just share an image here. So you can see that this is a eukaryotic cell cycle. And in this eukaryotic cell cycle, can you see my screen?
It just went off actually.
Okay, just hold on. Okay. Yeah. So basically, during cell cycle, what happens is we end up having various events, as I mentioned to you, that helps the cell to grow and divide. Okay. So this is an yeast cell cycle, which is represented here. You can see that there is a single cell which is there, and this single cell is in a G1 phase. That is just preparing itself to grow, replicate its chromosome, and divide. So at the end of the G1 phase, these cells basically enter into a phase called as S phase. At this S phase, what happens is that the chromosome inside the cell replicates into two different chromosomes, or depending upon the 2n number of chromosomes, and then And then these guys, what happened is that they end up segregating these chromosomes into the newly dividing daughter cells. So each of this cell, basically what you can see here, a small cell and a large cell, this is the budding yeast. And then you can see that this newly formed daughter cell will gain a chromosome during replication process. And this replication process happens during the M phase, both segregation and division. And during the M phase, what happens is that the newly replicated chromosome is pumped into these newly dividing daughter cells. And at the end of the day, it just divides to give new progenitor cells, newly born daughters. Okay. And so you can see that these various events are classified as different stages, and this comprises the cell cycle. And this just keeps on repeating so that the cells proliferate itself, multiply itself. Okay. So that's what mainly cell cycle means.
Before proceeding further, I would request our viewers to use the live chat option. You can ask any questions that comes to your mind and quench your curiosity, and Sunish will answer it. So Sunish is all yours today, and so we will proceed. Okay, so what's so different about bacterial cell cycle, Sunish?
Before that, the bigger question out there is most people ask is, does bacteria have cell cycle? Okay, so the larger understanding is that bacteria is just a sack of enzymes, boring unicellular organism which doesn't do any fancy things or which does not have anything complicated out there. Okay, but that's not the case. Indeed, research in the past 20 years, both cell biological and genetic experiments have shown that bacteria has proper cell cycle programs. And here, what I'm showing you is one of the most primary organisms that actually ended up
Okay, I think Sunish's
There's
network.
some network issue.
Yeah. So we'll wait for a while and let's see if it comes back. Great. So till then, since by the time Sunish checks his network, so the surprise. Since it's a final session of this season, I would like our viewers to know that we have completed like 14 sessions on across various fields, like in session 1, we started with an antibiotics session with Dr. Ashwin, and other session, like it was followed by Sunil session on yeast. It has this cool name, Breaking Bread and Other Breakthroughs. And in season 1, we completed like 7 sessions, and we culminated the season with Das's session on regeneration. And so in this other season, which is like the current season, the season 2, we started with different model organisms like Drosophila, and we even covered about plants and associated microbes. So we are a microbiology lab at NCBS. I mean, you can say that we are a microbiology lab at NCBS. Focus is bacterial genome and regulation. So we tend to have inclination toward bacteria more, and we will try to get back to different fields and bring researchers closer to you across different fields. So Akshara, any session that you like the most since you joined in season 2 majorly? Any session that?
Yeah, I liked the session conducted by Sandeep very much. It was very I gained a lot of knowledge from that session. Yeah,
Right.
it was very interesting for me.
Sandeep's session was on bacterial communication. And
yeah.
bacteria communicate. And
So it always fascinates me how these single cell organisms communicate with each other.
Sorry
okay,
Yeah.
about
so
that.
no problem, Sunish. We were
Yeah,
like
so
talking.
it's a home internet, isn't it? So it just behaves crazily
No,
sometimes.
no
So
problem.
yeah, so sorry everyone. What I was talking about is basically um, so this I'll try to explain this cell cycle using Colobacter crescentus as a model organism. The importance of Colobacter crescentus is that it produces cells with two different purposes. Okay, purpose of this cell is to swim around. This is an aquatic bacterium. It lives in fresh water. Purpose of this cell is to swim around and to find new nutrient-rich conditions wherein they can colonize and proliferate. Okay. So basically, this cell is not allowed to replicate until it finds a nutrient-rich condition. So they exist in a G1 like condition. So as I mentioned in the previous, as I showed in the previous slide, these guys basically exist in this G1 like condition. So now, and only the stalk cell basically gains its name from this appendage, which is present at one pole. And only this stalk cell is allowed to replicate its chromosome and divide. And this stalk cell is in an S phase like condition. Of course, they end up in G2M phases also. So once this G1 cell finds a nutrient-rich condition, what happens is that they enter into the stalk cell phase. That is, they shred the flagellum, they eject the flagellum, they retract these locomotory organelles called flagellum and pili, and then they produce the stalk at the same position. And the stalk at the tip has a gum, and using this gum, these cells can attach to solid surfaces and then keep on replicating its chromosome, dividing and producing new cells. Okay, and so this is here you can see that you have a G1 like cell, you have an S phase cell, you have a G1 to S transition, and you have a G2M phase. But if you ask me whether there is a G0 in this bacterial cell, in principle, there is no G0 because all these cells are highly active and they try to transition immediately. So, or the window of G0 is very small that we have not really looked into it yet. So now if you ask me whether this is unique to only this weird organism called Colobacter crescentus, that's not the case. It's also the case with pathogens like Pseudomonas aeruginosa. As you can see, Pseudomonas aeruginosa basically survives on the cell surfaces of the lung epithelium and primarily on the lung epithelium. And you can see that these cells basically swim around this planktonic cells, they swim around. And once they find a host cell surface, they switch to another form, which is called as a striker. Here, it's not a stalk which is produced to attach itself, it's a pili which is used to attach itself. And this striker then proliferates, basically giving out more spreaders and also several strikers in order to grow in a colony, which we will discuss it later. Okay, so this is what happens during bacterial cell cycle.
Sunish, as you told about this thing that bacteria generally do not have G naught stage. So, can we say that bacteria which are in dormancy, or like in case of Mycobacterium tuberculosis or other, which you happen to see in pathogenic bacteria?
Yeah. Very well. Because I don't say that there is no G0 phase, but there is no well-defined G0 phase. So absolutely, you can call those guys which are dormant to be in a G0 phase. For example, recent research from our lab has shown that, has also indicated that we can keep these Swama cells, the G1 cells, inactive for some time. So very well, that can be explained as a resting phase.
Also, are these like in eukaryotes, like the cell cycle that you mentioned, they are majorly dependent on the state of DNA content, right? Is replicating, so it's S phase, and in G1 phase, it's like when we studied in school, it's kind of preparing for the S phase and other things. So, so what marks these boundaries of these stages in bacteria?
Yeah. So if you mean to ask what triggers this boundary or what triggers entry into different cell cycle phases, isn't it? So in bacteria, we know, we exactly don't know what happens or at the molecular level, but there are several evidences that coming in together, which actually says that it might be the environmental factor which is triggering this entry into S phase. Okay, so the purpose of these cells is basically to go in search of nutrients. And until they find nutrient conditions, or in this case Pseudomonas aeruginosa, until they find a surface wherein they can colonize, they don't want to enter into the S phase. So there might be sensors which are sitting on the surface of the cells, which tells the bacteria, okay, you are in the right condition, now let's enter into the S phase. Now, what are the sensors? How the trigger is happening? Though there is some understanding of it, it's still preliminary. For example, even if you talk about Mycobacterium, the resting phase and the entry into the proliferative phase is still a field of less understanding, isn't it? Yeah.
So, Sunish, there is a question in the chat box, and Harini asks, so what do these bacteria do the cell cycle? Is it like using energy, food, or something else to maintain the cycle?
Yeah. Okay, so it's basically, as I mentioned to you, it's metabolism, isn't it? So I think so what happens is that there is a, just hold on, okay. Yeah, so there is a particular type of what we think, what we understand from our lab is that there is a particular type of signaling process that happens inside this cell, and there is another particular type of signaling process that happens in this stalk cell. Okay. So if you want me, I mean, let's say that I will explain it how I think it will be more easy to explain if I tell how these how the stalk cell becomes a stalk cell and swarmer cell becomes a swarmer cell, isn't it, or something like that. And and what we think is that there are certain metabolic processes, certain transcription processes or signaling mechanisms that happen in this particular cell, which keeps the cell in that particular state and does not allow the cells to enter into the next phase, for example. Okay, and of course that burns down again to lots of other questions. I mean, other regulators that operate here. Of course, most of the, I mean, we know a bunch of regulators, but as I mentioned to you, the understanding of all these regulators are not complete. Yeah.
Okay, and there's another question from Yogini, and she asks, do the sporulating bacteria also follow a similar kind of cell cycle pattern?
Yeah, so that's a good question. So what happens during so what is sporulation? So here in Colobacter, the cell cycle pattern is basically wired into the growth cycle, isn't it? So each, if it has to grow, it has to go through these processes. Okay, but in the case of sporulation, what happens is the bacteria enters into or produces endospores or spores as such only when they encounter drastic conditions like nutrient depletion or environmental factors which does not allow them to grow and proliferate and things like that. So you can, I mean, in a way you can say that spore is a resting cell which germinates and enters into cell cycle or enters into growth and division at a later stage. So as I was talking to Nitish before, as I was discussing with Nitish before, this particular spores may well be considered as a resting phase of a cell, but sporulation happens only in drastic conditions, okay, only in adverse conditions, and bacteria doesn't actually want to enter into all this sporulating or want to sporulate themselves because it's a highly energy consuming as well as time consuming process. Okay, and it doesn't what it says is that it doesn't allow them to proliferate as such, but it allow them to just enter into a phase wherein they can proliferate later when the conditions becomes favorable.
Yeah, that makes sense.
So Sunish, like in eukaryotes, as far as I remember from my school textbooks, there are some proteins which are like localized to some regions, and they kind of get modified, and then enter like the cell enters into some stage. So does that kind of localization you observe in bacterial cells also?
Yeah, absolutely. So, and that's a very nice question. So, what happens here is that bacteria, such I mean, you might have learned from your textbooks that it's just a single compartment, isn't it? There is no compartmentalization as such in bacteria. Okay, there is no proper compartmentalization as you see in the eukaryotic cells that happens in bacteria. But what happens is that these guys actually powerfully use protein production, localization of proteins, as well as signaling mechanisms in order to regulate all these things. Okay, so protein localization, as you pointed out, indeed bacteria localize proteins. For example, here you can see that this is some images from my lab. So here you can see that this is GFP. This is a flagellar protein which is localized to one pole because the flagellum is present only at one pole here, isn't it? If flagellum is only present here, and so the protein needed to make this flagellum, they localize only to one pole. Okay, and same is the case here. This is a protein which is used for asymmetric or cell-fate determination in bacteria, and that's again localized only to one pole. And these localization of protein again is in a hierarchical manner. Okay, and these proteins are localized not always, but most probably during certain stages of the cell cycle, because it might be a localization dependent activation, and only upon localization they become activated, and then they might be bringing forth the function of this particular protein. Okay, so for example, what happens is that this red color protein which I'm showing here, this localizes only during the mid S phase in Colobacter cell cycle, and here you can see that this protein is mostly throughout the cell, and this is the Colobacter cell shown under DIC, and you can see that the fluorescence is throughout the cell. Okay, but as the time progresses, you can see and this is the start. This is a G1 cell, and this cell is basically entering into the S phase. And once they progress in S phase, as the progression happens, you can see that the fluorescence from the cell body decreases, but you start getting this kinds of pointed localization or pointed foci at one tip of the cell. Okay, and so here what happens is that the function of this particular protein is needed only at this time window, and that's why they're produced early. Of course, there might be a different function for this protein when they are delocalized. We don't know about that, but what happens is that these guys are when these guys are localized, they trigger a signaling mechanism. Okay, and that's one mechanism. So they produce proteins, and they sequester these proteins to specific regions in the cell, so that it's not spread throughout, and the function of this protein is delimited to only one particular region in the cell. So that of course localization happens, and protein localization happens in bacteria. Not only protein localization, and again, this protein localization, as I mentioned to you, this is brought about by a cascade of events. Okay, and for example, here what I'm showing you here is that during the G1 to S transition, you are localizing one protein first, that is the first regulator first, and once this first regulator localizes, then it regulates it brings in the second regulator of the cell cycle process, for example, if you want. And then once the second regulator is localized, they bring in the third regulator. Okay, so hierarchical dependent localization happens, and also nowadays there is another evidence which shows that the new in thing, which is phase separation, which also operates in all these localization processes. Also, these protein localization also depends upon where your chromosome is. Sometimes these proteins don't like to go to regions where the chromosome is present, and they just try to avoid the chromosome, and then they try to, and they use this process in order to find where they have to localize. So, for example, in this cell, the chromosome might be spread throughout the cell, but they might not be present at the poles, and that might be the reason why these are localized at one pole. Okay. And not only localization, in eukaryotic cells, you might have studied from your textbook that some proteins are produced only during specific stages of cell cycle, and that also happens in bacteria. And here I'm just showing a cartoon of protein A and protein B, and you can see that this protein A and B are produced are present only in the G1 cells or in the G2M cells or late S phase cells. and they are not present in the mid-S phase cells. And this control either happens at the level of production, basically transcription and translation, and also it happens at the level of proteolysis. So there are proteases that are present in bacteria that chew up these proteins and delimit these proteins only to certain stages in cell cycle. Okay, and this has been well studied in bugs like Colobacter and Bacillus subtilis, and of course nowadays with asymmetry being seen in even bugs like Pseudomonas aeruginosa and Mycobacterium, these kinds of processes are also being studied there.
Okay. Yeah, Sunish, I have a question. So in eukaryotic cell cycle, we have checkpoints, right? So is there, like, can we consider these proteins as a checkpoint for the prokaryotic cycle?
Absolutely. Yeah, that's a fantastic question. Yeah, indeed. So what happens is that this A protein, for example, if it is not cleared, then the cells don't enter into S phase. So it is very much important that this particular protein is cleared off. Now, what triggers this clearing is basically activation of a protease, okay, and a signaling mechanism which ends up in activating a protease. And if this protease is not activated, these cells stay at a particular stage itself. Again, there are secondary messengers like cyclic di-GMP and things like that, which are also important for this transition. And the levels of cyclic di-GMP goes up and down. The secondary messengers go up and down during these cell cycle stages. And if you manipulate this cyclic di-GMP levels also, you can end up inhibiting cell cycle, and that acts like a checkpoint. And nutritional condition also acts as a checkpoint. Okay, so but again, at the molecular level, it might be these proteins which are acting, and the nutrition condition might be feeding onto these kinds of proteins. Yeah.
Okay.
So just to simplify things, like whatever you just said, is like bacteria can regulate the protein levels in the cell to move, like to transition to different stages, and it can do by increasing the production and even decreasing the protein content by some proteolytic enzymes, right?
Yeah, so basically what I'm saying is that if you want to produce a protein, the entire code for producing this protein is basically present in the DNA, isn't it? So the DNA is transcribed into RNA, RNA is translated to make proteins. So now this cell might have made the protein, but now if this cell has to transition into this S phase cell, then you need to remove this protein. So switching off transcription and translation is not enough. You need to also physically remove the protein. So how do you remove that? So that's when the proteases come in. The proteases basically chew up the proteins which are already present inside the cell. So that is why you have, and it's amazing. Bacteria might be because it's a unicellular, it's uncompartmentalized. Well, that's what they say. So because of that, these guys have several layers of regulation. It's not just transcription, translatory regulation, not only proteolysis. It also uses a third level of regulation like signaling mechanisms, sensors, and things like that. Yeah.
Yeah, in line of these sensors, there is a question from a group called Little World. What is the type of chemical signals that these bacteria are looking for? Is it is some food source or some other kind of chemical signal where, like, basically when the spreader decides to become stationary and start dividing, right?
Yeah.
So is there a particular molecule that triggers this?
Yeah, that's a fantastic question. We actually don't know what is the exact molecule, exact environmental molecule that triggers this. Of course, if it's a Pseudomonas aeruginosa, it might be sensing something on the cell surfaces, but what it exactly senses is we still don't know. Again, in case of Colobacter, which is more easier to study in the lab, what we have done in our lab itself is that we have taken this form. So an interesting aspect of this, why we use Colobacter to study this cell cycle processes is basically we can synchronize this G1 cells. We can just isolate this G1 population and study this in a synchronized manner. So when we have isolated this G1 population and resuspend them into a nutrient-rich condition, they immediately enter into S phase. Okay, so then the same question came to our mind: which nutrient is actually important for this transition into S phase? So what we did is we just used a minimal media, and then we started just playing around with different kinds of nutrient conditions. And of course, we have a preliminary understanding, but we are not still sure to say that certain nutrients are very much important for this guy to transition, okay, into S phase, okay. And yeah, but to answer that question exactly, I think more research is needed, and we are still hunting for what is the exact nutrient trigger or the signal that bacteria perceives to transition into a stalk cell or a striker cell. Yeah.
Okay. Yeah, there's another question from Farheen Anwar. How do cells know that there are no more damage to its genetic material or other damages that might have happened during the preparation of division to enter into next phase of cell cycle?
Yeah, so of course, when there are, as you had asked me whether there are checkpoints, there are proteins, checkpoint proteins, which actually try to, which are related to chromosomal damage. So if there is a chromosomal damage, or if there is a damage to the chromosome, or there is no proper, what do you say, replication event that has been completed, then these cells don't enter into S phase, okay? They or don't proliferate at all. And so then at that time, what happens is that these checkpoint proteins, the sensing of these sensory proteins trigger the checkpoint proteins, and these checkpoint proteins ensure that they don't enter into the S phase. So one example, which I don't have images here, I had to pull out some slides for that, but a recent example which we came out, which we have just shown from the lab, is that that making of this flagellum. So when these daughters are made, it's important that the flagellum, it's important that one of these daughters are born with the flagellum, okay, because the function is to swim out, isn't it? So now what we saw in the lab is that if you stop making of this flagellum, if you interfere with assembling the flagellar components, then the cells don't divide at all. They don't enter into the G2M phase, okay. So and we actually describe the protein which actually links, which senses this flagellar assembly and links to cell division, okay. So that's one type of checkpoint protein. Another thing which we have also observed in the lab is that so bacterial chromosomes are circular, isn't it? And when they replicate, they end up having these catenated chromosomes, okay, the interlinked circles. And now these catenated chromosomes are resolved by topoisomerases. Topo4 is specifically the enzyme which ends up resolving this. And when we were studying some other proteins in the lab, we also found that if this resolution does not happen, if you inhibit this topoisomerase 4 and don't allow the chromosome to decatenate, then also they arrest in cell cycle. Okay, and some checkpoint proteins are triggered, and then the progress in cell cycle does not happen. So there are sensor molecules which can, there are sensor proteins which can sense these kinds of events, developmental events, including replication, flagellar biogenesis, and things like that, which can couple progression, cell cycle progression and proliferation.
So, Sunish, there is a lot of mobilization of these proteins happening within the cell, and they're assembling at from one point to another and moving there. So, there's a question from Adav in the chat box. So, how do these proteins know where they have to go, and what is the kind of signal that takes them to their destination?
Exactly. So now I will explain this with how. So you need to place the flagellum here, isn't it? At this particular point, at this particular pole. In fact, I should have also put that cartoon there, because those are really interesting questions. So now what happens? So now this is one pole at which this flagellum is positioned. Okay. Now, what was this pole? this pole was previously a cell division plane, isn't it? This is where the cell division happened. So now the cell positions this flagellum at this particular pole by hunchbacking on the cell division apparatus. So the information to place the flagellum at this new pole. So when this divides off, these two are the new poles, and this is where the new flagellum has to be made, isn't it? So the information for that is actually placed at this division plane by hunchbacking this bird scar protein, if you want, or this marker onto the divisome. So when the cell divides, then both the daughters at these new poles inherit that particular protein. And then they know that, okay, this particular flagellum has to be placed there in the next cycle. Okay, that's one example. Now you might be asking how the divisome itself is localized. So there, what happens is that now this chromosome is replicating. Now that when the chromosome replicates, what happens is that you have origins in the chromosome, and these origins are the ones which where the initiation of replication happens. So in Colobacter, what happens is that once these origins replicate, they are immediately tethered to the opposite pole. And bound to this origin is a protein which basically localizes at both poles. So you have one origin here and the other origin here, and this will end up forming a gradient. Okay, so there will be high concentration of protein at one pole because these are bound to the origin, and that will actually release a gradient towards the center. So now this gradient will decrease at one point. There will not be any of this particular protein at one point, and that's where the cell decides to divide. That's where the cell can assemble the divisome and then divide. Okay, I'm sorry, I don't have that slide here, but it might take some time for me to dig that out. Yeah.
So that related to similar thing, like, okay, you told that divisome somehow dictate where to divide and other things. But does bacteria have sense, like bacterial molecules? Like I'll just ask a big question, like, does bacterial molecules like proteins and other things have sense of location or coordinates in them? And
you mean to say that where they are,
where they are, like how do they know that, okay, this is the end and this is the middle? And
okay, yeah. So so one thing which and one of the idea which is out there is basically the curvature. Okay, so now this is a negatively curved portion, and this curvature can be used as a sensor in order to localize these kinds of proteins. So at the negative curvature, these proteins are basically localized. Okay, and that's one way of localizing it. Another way, one of my favorite models is that now this is a gram-negative bacterium, and you have membranes, you have cellular membranes, and inner membrane, peptidoglycan, and outer membrane. So there might be signatures in this membrane itself, or chemical modifications in the membrane itself, which these proteins might be recognizing and localizing at that particular position. Okay, in this case, for example, regulator 1 might be something which recognizes a signature in the membrane or in the peptidoglycan itself, and this particular signature might be present only at one pole. And once the regulator 1 is localized, then it's easy to localize regulator 2 and 3 because they have to go to regulator 1, isn't it? So the cue might be given by these curvatures, or the cue might be given by chemical status or the physical status of the cell itself. Okay.
So we have a couple of questions from Harini in the chat box, and the questions are, they are quite existential to cell cycle in general. So does cell cycle, so the question is, does the bacteria wantedly do the cell cycle? And if yes or no, then why?
Okay, so for example, in this case, or in Pseudomonas aeruginosa's case, or in Chlamydia trachomatis case, it is better. It needs to have a cell cycle program because it needs to produce a type of cell which can go and colonize a new condition, or go and infect a new host, as well as entry into the host cell and things like that. Let's not get into that complication. Okay, in certain bacteria, of course, you need this. You need to have this patterned cell cycle, but you can interfere with them, and then you can bypass these things. But then what happens is that most of these bacteria are compromised in their are slow growing, or they compromise in their growth and proliferation time and things like that. But certain cells, for example, E. coli and things, they don't have a pattern cell cycle. Everything is overlapping, isn't it? So I mean, basically what happens? So E. coli divides faster when you're growing E. coli in a nutrient-rich condition. What happens is that they have to divide. They divide at 20 minutes. So now, before they divide, the chromosome has to replicate. Now, the chromosome replicates slowly. So in order to compensate for that, what happens is that the daughters inherit a completely replicated as well as a half replicated chromosome, isn't it? So in those cases, what happens is that this entire thing is interlinked, but still there is a replication process that has to happen. There is a division process that has to happen, and there is a resting process that has to happen. And if all these things are there, then you have cell cycle, isn't it? So without undergoing chromosome replication and division, they cannot proliferate. So what it says is that without cell cycle, they cannot proliferate.
So yeah, in continuation to that question, yeah, the question being asked by Harini is, does cell cycle help bacteria to survive under conditions, or does it help in survival?
Yeah, of course. Yeah, definitely. So in this case, so what is survival? You need to find new host to proliferate and to live, isn't it? So of course, this cell cycle process in these conditions, it helps the bacteria to survive. And as I mentioned to you, replication and division is interlinked, is a cell cycle process. And without replicating and division, they might be a cell just existent for some period of time. But if they want to proliferate, they definitely have to undergo cell cycle. Yeah.
Amazing. So we had like great questions, and let's proceed with. So I'll ask another question about how, as you told, that bacteria had some signaling mechanism. So what kind of signaling happens in bacteria?
Okay, so there are so basically this signaling happens because of protein-protein interactions, or there might be chemical modifications that happens in this protein, which helps a protein to get activated. Okay, and that basically this signaling process tells the protein when to get activated and where to get activated. So I think, let me see. I can explain this with an example of phosphorylation, okay, which is easy to explain. So now when you add a phosphate to an amino acid, so in this case, it's a histidine which is getting phosphorylated, and an aspartate which is also getting phosphorylated. So, okay. So now let's say that you want to have a sensor. There is a sensor which is sitting at the outer membrane of the cell, and the sensor is the one which the bacteria uses to decide the environmental condition or sense the environmental condition. So in this case, what happens is that this sensor is a kinase, okay? And once this sensor is activated, it just takes a phosphate from an ATP and adds this phosphate onto the histidine, okay? And in the kinase domain. And this phosphate is transferred onto another protein, which we call it as a response regulator. And only when this response regulator is phosphorylated, it becomes active. If this response regulator is not phosphorylated, it will not become active. And when does this response regulator becomes phosphorylated? Only when the sensor kinase gets activated. And when does the sensor kinase get activated? Only when they end up encountering a particular environmental condition in which this particular response needs to be activated. Okay, and again, this transfer of signal via phosphate, or this signaling via phosphate, might be as simple as just one sensor kinase which gets phosphorylated and then adds a phosphate to a response regulator, or it might be a series of events wherein there are several intermediate proteins which are involved in this relay, and then you get a larger signaling network activated. I'll just explain this using how two types of cells are made in our favorite organism, basically Colobacter crucentus again. Okay, so now there is a protein. Let's call this protein as sulfate determinant protein. Okay, and when the sulfate determinant protein is phosphorylated, the cell becomes a stalk cell. When it is not phosphorylated, the cell becomes a swarmer cell. Okay? So now, in order to add
I had one question regarding the signaling mechanism that you just talked about.
Mm-hmm.
So, from the figure, it looks like there is some molecule which is binding to the sensor kinase, which in turn sort of signals the.
Oh, this orange, the orange one?
No, above, outside the membrane, the signal. What
yeah.
kind of signal is that? Is
So
it
this might be anything. This
something?
might be food. This might be an environmental signal. This might be a trigger. For example, host cell surface triggers from the host cell. Anything. So it depends upon. It's not just one kinase which is sitting in bacteria. There are several different kinases, and each of these kinases might be sensing different things.
Yeah. So, for example, we see that bacteria can alter their physiology on the basis of how much oxygen is present in its environment. So, if there is a hypoxic condition, how can the sensor kinase sense that kind of a signal? So, what kind of molecules do bind to this?
So basically, there might be sensor kinases or sensors which are sitting in the bacterial membrane or even inside the cell, which basically triggers a signaling process. And it need not be a phosphate dependent signal. I'm just giving an example that it is a phosphate dependent signaling process. It might also be a different type of signaling process wherein proteins are modified by cysteine disulfide bonds. For example, when there is a oxidative, hyperoxidation happening inside a cytoplasm. So there is an oxidative stress that happens, isn't it? So when this oxidative stress happens, there is a molecule which is called as OxyR which gets activated. And this OxyR is a stress regulator, and it needs to be activated only when there is an oxidative stress. And this OxyR gets activated because of cysteine disulfides. And the cysteine disulfide forms only in oxidized conditions, isn't it? So, and that is again, that's a signaling mechanism, which, and then once this OxyR gets activated, it's a transcription factor. And they turn on a plethora of proteins or turn on a plethora of genes, which produces the proteins which can help in decreasing the oxidative stress. So again, like the previous question, like what is the signal? You cannot say that it is just one signal which is activated. Okay, so these kinases might be sensing various kinds of things. Okay. So again, so how this phosphorylation works, how this phosphorylation works, just I'll explain it using the sulfate determinant. As I told you, this sulfate determinant is phosphorylated in one compartment and dephosphorylated in another compartment. Now, if you want to add a phosphate, what do you need? You need a kinase. So what happens, again, if you remember protein localization, there is a kinase which is localized at one pole of the cell. And this kinase, basically, the job of this kinase is to add phosphate to this sulfate determinant. And there is another phosphatase which is localized at the opposite pole. And the job of this phosphatase is to remove the phosphate from this sulfate determinant. So now in an undivided cell, what happens is that both this phosphatase and the kinase are present in the same cell, maintaining an equilibrium of phosphorylated and non-phosphorylated form of the sulfate determinant, isn't it? So, but once the cells divide, what happens? The phosphatase is compartmentalized only to one compartment, and the kinase is basically sequestered to another compartment. And because of this, there is complete dephosphorylation that happens in one compartment and complete phosphorylation that happens in another compartment. And this triggers the signaling event. So this triggers the complete dephosphorylation of the sulfate determinant, triggers a cascade of signaling events that starts the swarmer cell program, or the cell to makes the cell to be a swarmer cell. And once the sulfate determinant becomes completely phosphorylated, what happens is that it triggers a stop cell program, a cascade of signals, which ends up making the cells to replicate and divide. Okay. So so these are the conditions. So this is an example of how this kind of phosphorylation signals or two component signaling system can end up in this case determining the type of the cell that is being born, which daughter is to be the swarmer and which daughter is to be the stop. But again, this signal can, as I was mentioning, this signal can also be used to sense molecules. Nutrient-rich nutrient molecules. They can be used to sense various kinds of stuff. Okay, various kinds of things that happen. Yeah, even host cell surfaces. Suppose if a bacteria is landing on a host cell surface, then you need to inject. The bacteria uses some some kind of injection needles in order to inject some virulent factors into the host cell, and these triggers also might be happening. And these sensor kinases might also be sensing those signals. Yeah.
So, I mean, I have this question which actually is asked by Harini also in the live chat. box, that how do you get to know this? Like bacteria are so tiny, and then you showed some colorful proteins and other things. So how do you like get to know all this about that tiny bacteria?
Yeah. So now once we find a protein, okay. So now if you want to study localization, what happens is that you just tag these proteins to GFP. For example, this is Flyg. Okay. Now we know that previous work, 20 years of work has shown that this Flyg is basically involved in making flagellum. Okay. So now if you where is flagellum made? So now flagellum is made only at one pole, isn't it, in this cell? So now what we did is we just tagged this Flyg with the GFP at the C terminus. And now once the Flyg GFP is there, what happens? The fluorescence will come, the GFP fluorescence will come only where Flyg is present, only where this particular protein is present. Otherwise, it's not. That's one cell biological mechanism. Another way to our favorite method of investigating this is genetics. We just use various genetic methods like suppressor mutations, random mutagenesis, interaction studies, and even we use biochemical interaction studies in order to find which protein interacts with what. For example, let's take this case of Flyg itself. So now I want to know which protein this Flyg is interacting with. So what you do is either you use a GFP, you take this Flyg GFP itself, you use an antibody against GFP, and then you can pull down Flyg, or you can immunoprecipitate Flyg from live cells, and then study what are the proteins that are coming down with this protein. Okay, so if a protein is X protein is interacting with Flyg, then this X protein will come along with Flyg. You can use either, if you are taking an educated guess, then you can use immunoblots and or various analysis in order to find what type of protein is interacting with them. But you can also use mass spec approaches in order to find what are the proteins which are interacting with this. So now let's say that another thing, we don't know what brings Flyg to that particular pole. Okay, so then what you do is you just take this Flyg cell, which is localizing GFP, you randomly mutate this cell. So now look for cells which does not localize this Flyg, which does not have this foci for Flyg. So that means that you have mutated a component which is required for localizing Flyg at that particular position, isn't it? So these are various methods. So cell biology using these fluorescent proteins, biochemistry like mass spectrometry, and genetics can be used in order to understand how this entire process happens.
Yeah, actually, we discussed about genetic approaches in session with Deepti Trivedi. Talked about Drosophila genetics and other things.
Fantastic.
And yeah, great. So, Sunish, can you go back to the slide that you showed, the flagella and like, yeah, no, yes,
The
yes, yes.
phosphorylation.
So what are these two things, like what this curvy thing is a flagella, I think, and then what are these two strands which are like?
Oh, these are pili. So these are pili. So what happens is that either these pili can be used for twitching motility, or they can also be, in this case, they are used for twitching motility, as well as they are also used as sensors. So this guy just swims around, and then once it hits a nutrient-rich condition, now they have to produce the stock only when they hit a solid surface, isn't it? So there's a beautiful paper which is out there which shows from E. Brown's lab as well as Ursinal's lab, which basically shows that this pili is used as a sensor, and then this sensor also triggers the sensing of this pili of solid surfaces, also triggers the cells to enter into S phase. Okay, so and these are basically used for twitching motility, and these are like these are type 3 pillars. And you might have heard about sex pillars also. Sex pillars are the ones which are used to transfer DNA from one donor to a recipient during biparental mating or conjugation. Isn't it? So those are the structures which are out there.
So I had a follow-up question on that. So we see that these stalks have some gummy structure on the top of it. So what
Yeah.
kind of chemical composition is there in this gum?
Yeah, so these are basically sugars, exopolysaccharides, but I don't remember the exact composition on the top of my head. But of course, the composition of this gum has been delineated just recently, and this gum is basically used to attach to solid surfaces. As I told you, this pillars can actually sense this surfaces, and once this is triggered, then the stalk is produced, and immediately the gum is produced, using which the cell attaches to that. Because and then this gum is very strong. Indeed, it is the strongest gum, biological gum, that man has ever known. What they say is that a drop of this gum is enough to is so strong enough that it can lift an elephant. So it's so strong. Okay, so yeah. And so that is so this gum is used for this bacteria to attach to streams, and then help them not to get dislocated and things like that.
I see. Yeah. So I was wondering if the cell membrane is of similar composition as the gum is, or is it just? Oh, I see.
No. Yeah. So the stalk itself is an extension of the cell membrane. So the stalk also has inner membrane, outer membrane, and peptidoglycan, but the gum is of a different nature.
Yeah. I see.
So there are various genes, there are various proteins that are segregated into the stalk, and that is actually used to make this gum, and then secrete this gum only at that particular position.
That's interesting.
Yeah.
Yeah. So there is a question from Yogini. What happens when a cell enters stationary phase of growth? And is there any correlation between bacterial cell cycle and the production of secondary metabolites?
Okay, it depends upon what type of secondary metabolites you are looking at. Okay, so of course, secondary metabolites are produced at various stages of cell cycle, isn't it? If you look at cell cycle, or if you look at a growth curve in bacteria, some secondary metabolites are produced during the log phase itself, some are produced during the stationary phase, and some are produced during the late stationary phase. Okay, so in a stationary phase, normally what happens is that early stationary phase, what happens? So basically the growth and death is almost the same. There is an equilibrium which is attained, isn't it? So after that, what happens is that there is a nutrient depletion, and it depends upon what type of bacteria you're looking at. Suppose if it's a Bacillus subtilis or any sporulating bacteria, then once the nutrients gets depleted, then these cells trigger the sporulation mechanisms and enter into sporulation. Okay, and produce spores, so that they wait until the nutrient-rich conditions comes in, and then they can grow and proliferate. Some of the bacteria, what happens is that, like Colobacter crescentus, what happens is that they just turn off most of the cellular events. Okay, cell cycle as well as growth events, and then probably we don't know whether it's exactly a resting phase or anything, but probably when you transform them into a nutrient-rich condition, they can regrow itself. But secondary metabolite production, it need not be only in the stationary phase that you get secondary metabolites. It can be produced at different stages of cell cycle.
So there's another question from Fareen, which is a follow-up question to his this DNA damage thing.
Okay.
Yeah. So he wanted to know, how does the cell know that the damages have been repaired and it is safe to continue the cell cycle? Basically, what switches off the checkpoints?
Okay, so so if I get into the details of what are the exact proteins that work on this processes, it might be like another session, and it might be too complicated. So what happens is that, of course, if there are damages that are happening, so what happens? If there is too much of damage which is actually happening, then the cells enter into an SOS phase, isn't it? So DNA damage checkpoint is triggered, and then cells enter into SOS, and the SOS protein stops cell division, and then they wait until entire damage is cleared off. So what happens? So the cells sense that there are no more single-stranded DNA which is floating around, and then once this is happening, then all these proteins which are, for example, SOS dependent, are cleared off from the cell, and then your proliferation starts. So when this SOS response is triggered, what happens is that concomitantly the proteins proteins which are used for proliferation and division are downregulated. Okay, because you want to invest all your energy in order to repair the damage. Okay, and there are proteins which sense this whether the damage is repaired or not, and after that they can enter into cell cycle. So they just turn off the process and then turn off the response process and then enter into the cell cycle. Okay, if you want me to go further, so what happens is that so now, as I told you, so these phosphor signaling mechanisms not only help the cells in order to proliferate and things like that, they also help the cells to find their way. Okay, so now when you have a swimming cell, so for example, there's a movie which actually shows cells swimming around, bacterial cells swimming around because of their flagella, and you can see that they are finding their way. They are just turning, tumbling, rotating, and all those things, and then finding their way. Again, this phosphor signaling mechanism, which I was talking about, comes into play to tell the cell whether they are to swim forward, whether they are to swim back, and things like that. And now, the purpose of this cell, as I told you, is to find nutrient-rich conditions, isn't it? Is to find where your nutrients are. And this is actually sensed by an apparatus which is called as a chemotaxis apparatus. Okay, so now this what is this chemotaxis apparatus? Is basically it's a bunch of sensors which are loaded onto the cell, mostly on the poles of the cell. Okay, and these sensors can sense the presence of food or presence of unfavorable agents, and then relay it to the bacterial flagellar machinery. Okay, and this relay also happens through this phosphosignaling mechanism. Okay, so for example, now in this case, the sensor is actually triggering a kinase, and this kinase is phosphorylating the response regulator, which is actually telling the flagellum to change the rotation. So now, if the flagellum is rotating in an anti-clockwise direction, the cells will move forward. But at the same time, if you just immediately change the rotation, then they will tumble or they will reverse the direction, isn't it? And so this is what happens. So the sensors will trigger an array of phosphosignaling or phosphotransfer events, which basically tells the cell to tumble or change directions and things like that. Okay, so how this and this is how this entire this is how the bacteria find their way to where they want to go. Again, why I'm showing this is just I'm showing you as an example of another example of how signaling mechanisms, for example, phosphosignaling mechanism again here can be used for bacteria to find their food source, okay, or find its way in the liquid medium. Okay.
Great. I don't know. So you talked about this sulfate in choice in Colobacter, right? So is it similar to differentiation in eukaryotes, like stem cells get differentiated?
Yeah, I mean, at base, I mean, so it uses a conceptually similar process. For example, as I told you, it's a localization of a kinase and a phosphatase at two different poles, which helps to phosphorylate and dephosphorylate sulfate determinant molecules, which helps the cell to proliferate or differentiate, isn't it? So in the and that's what is happening. For example, if you look at neuroblasts, Drosophila neuroblasts. So what happens is that you have this protein called as LGL, lethal giant larval protein, and this is the sulfate determinant there. And the phosphorylation or the dephosphorylation status of this LGL protein will tell you whether will trigger this asymmetric division, okay, in these cells. So it's conceptually similar, but it's not the same proteins which operates in this process.
Right. So I mean, it's so amazing that in that small tiny cell, like everything is happening, and it is able to regulate. And we like there has been a research on bacteria for quite a long time, and we still have a vast uh, maybe world to explore.
Yeah, and the events are so well coordinated. It's really amazing to see that in a very
Yeah.
small system.
No, so the amazing thing is that though it appears like a small, simple cell, the signaling events and the preciseness in which they coordinate this localization of proteins and activation of proteins is amazing. Now, if you look at the volume of a cell, bacterial cell, it be just four femtoliters, for example. And this in this 4 femtoliter volume, basically it's just coordinating all these events, isn't it? And so that's really amazing. It's something which has always amazed me, and that's something which led me to investigate this process and or use bacteria as the model system to study these things.
Yeah.
And of course, these are important events in bacteria. It's not just the fundamental knowledge which you're gaining from this. These might be in future. These might be the molecules which you can exploit as drug targets, isn't it?
Yes.
Because you're studying important regulatory events in bacteria which stop them at different stages in cell cycle or proliferation. And then so now if you understand this signaling mechanisms very well, then probably you can end up choosing, or not probably, already people are using molecules in the signaling mechanisms as drug target molecules. Okay, so and that's an amazing thing that has been happening. So another thing is that, so now someone is asking me, like, how does cells, okay, cells have found the food source. So now how do they relay it? So phosphosignaling mechanism is one thing. Another thing is metabolism. So metabolism can also be used as a signaling mechanism, isn't it? So there might be a particular type of metabolism that is happening when you have a particular food source, whereas a different type of metabolism might be happening when you have a different type of food source, isn't it? So, for example, what I can show you here is that so the swarmer cell, wherein a different type of metabolism is happening, you have a reduced cytoplasm because it's produced by a particular food source, and then you have an oxidized cytoplasm which is produced by a different type of food source. And again, that sensing, so that's how not only the availability of the food, but the metabolism is also integrated into the developmental event. And it's very important that metabolism is properly integrated into the developmental events. Otherwise, that's going to be detrimental for any organism, isn't it? Because it's not going to, if you're not going to fine tune your metabolism based upon the food source available, then it's not going to be beneficial for any of these organisms, isn't it? So, and so
I always
now,
thought
yeah.
cytoplasm was only reducing. So this
Sorry?
is something very new that I'm here. Yeah, so I thought that the cytoplasm was always reducing in nature, but this is something very new that I encountered just now.
Yeah. So absolutely true. So I mean, we hit upon this. So this is something which we reported some time back. So what we saw is that we were working with a protein, and that protein basically gets activated during using cysteine disulfides. Okay, and now in a normal cell, and this was a cytoplasmic protein, and if you want to activate this protein using cysteine disulfides, then the cytoplasm needs to be oxidized. So that's when we went ahead and investigated whether the cytoplasm is oxidized. And indeed, to our surprise, we saw that the cytoplasm is indeed oxidized. And then they can be used for, this probably, we are still to find out what's the exact metabolism which operates this. But this is linked to metabolic events. Okay, and so there is a specific type of metabolism that might be happening here, output of which basically oxidizes the cytoplasm. Okay, and we also went ahead and played around with this. So we took this cell, we stopped the oxidation using chemicals, and then you can stop the G1 to S transition. And we took this cell, and then we allowed the cell to progress in oxidation, but we stopped the oxidation to reduction transition, and then again you can stop them at this position. Yeah, in fact, this is the first one of the first reports which shows that cytoplasm of the cells can be oxidized. And together with this, there are also recent evidences which shows that this happens in pathogenic bacteria too. Okay, so the pathogenic bacteria have these type of G1 cells which only infect a host cell, and then they also might be playing around with the cytoplasmic redox in order to survive.
So Yeah. So does this oxidation happen throughout the bacterial cytoplasm, or is it locally present in some places where the
Yeah,
um,
so as far as we think that it is happening throughout the cytoplasm, but again, in order to exactly understand that, we need to do some microscopic studies, which we are in the process of doing it. So that will help us to understand whether, suppose if this cell is dividing here, whether you have a oxidized, more reduced compartment, more reduced cytoplasm here and more oxidized cytoplasm here, we still don't know. So that's what we are trying to find out.
yeah,
So
certain
these for
gradient
this,
would be interesting if it exists.
absolutely, absolutely, because for this single cell studies are required, and that's what we are doing now. So we need to design a special sensors in order to really delineate this, tease this out. Okay, so again, what I'm saying, what I'm trying to tell you here is it's not only phosphorylation. It is another, this is another layer of regulation that is basically happening. So bacteria is so complicated that they have, if one mechanism fails, they have another mechanism to stand in for it. Okay, so they just have multiple layers of regulatory events which basically end up fine tuning this entire process. Okay, so once the cell, so what happens once the cell hits a cell surface? Okay, so for example, I'll explain it using Pseudomonas aeruginosa. So now let's say that this is a spreader cell, and then this is going around finding a nutrient-rich condition. So once they hit this cell surface, what happens is that they just try to proliferate there. They don't want to. They have found the host cell surface. They just stick themselves to this host cell surface, and then they will produce many of the cells, and these cells will be held together by this matrix called as biofilm. Okay, and when these cells are held together by this matrix called as matrix, like biofilm matrix, not only does this help this bacteria to live in a kind of a colony, or in a semi multicellular environment, but also this helps the bacteria to protect themselves from antibiotics. So for example, this is a biofilm. Now, if you're treating this biofilm with antibiotic, you're trying to add antibiotic and kill this bacteria. So antibiotics it's very hard for antibiotics to penetrate. So only the bacteria which is residing on the surface of this biofilm might be killed, but the rest of these guys which are inside this biofilm might not be killed. Okay, so they might be protected against this antibiotic instance. So biofilm formation and not only helps bacteria to invade and proliferate on the host cell surface, it also helps this bacteria to protect themselves from various environmental factors, including antibiotics. Okay, and once they deplete a nutrient, so you have a cellular damage, the host cell is completely damaged, they have eaten, they have fed on all the nutrients which is available, then what happens is that this biofilm just dissociates itself, and this swimmers will be propagated, and then they will go and colonize another host cell, and then they will start dividing and producing a biofilm at the same position. Okay, yeah.
So since we are like we have almost we got to know that this bacteria are like single cells, though they are single cells, and but they can also exist in like a big community like structure, like in biofilms. And you also talked about how this bacteria decides cell fates, right? So I mean, they both are like what we thought that it happened in eukaryotes. So I'll just ask a question that just like in eukaryotes, do they have some kind of circadian rhythms also in bacteria?
Yeah. So circadian rhythm, for example, in these, let's say that in these things, you need to. So if you want to form a biofilm, you need to coordinate yourself in this biofilm, isn't it? All the bacteria which exist in this biofilm needs to speak the same language. Isn't it? They need to coordinate themselves. So, of course, quorum sensing is one mechanism by which this coordination happens. Okay, I think you guys might have discussed quorum sensing in previous sessions.
Yeah,
So,
in Sandeep's session on bacterial communications.
yeah. So, and coming to circadian clocks, that's another interesting aspect. So that also a trick which bacteria can do. Cyanobacteria, for example, they have the circadian rhythms. For example, this is a circadian clock represented in cyanobacteria. It's a it's a fantastic recent paper which came out. So here you can see that this yellow protein is turned off at this position. Okay, and this just gets turned off and on in a very synchronized manner. All the cells are producing this yellow protein, then they are switching off, switching on, switching off, switching on. And you can see that this particular rhythm is happening. Okay, this kind of day-night rhythm is happening. And if you want to see it in a movie from the same paper, you can see that. So what you're seeing on the left is basically the cell is shown by this red fluorescence. But what you have to look at is this green color protein. You can see that over a period of time, as the cells grows. Now this is a chain of multi-cells, isn't it? There's a cyanobacteria, and there are several cells here. Each of these cells are interlinked in this chain. So now you can see that all these cells turn on the green protein at the same time and turn off the green protein at the same time. So you can, okay. Yeah, so this is, you can see that, now the green protein comes on, it switches off, comes on, it switches off. Okay, and this coordination, again, this coordination is achieved by circadian clocks. Okay, whereas the other coordination wherein I showed that biofilm formation and things like that happen, so you need to talk with each other, there it is achieved by quorum sensing. Of course, circadian clocks and circadian rhythms happen in bacteria. For example, cyanobacteria is a fantastic example for this.
Yeah, so like circadian rhythms regulate multiple processes in eukaryotes, right? So are there any biological processes known which like these kinds of rhythm regulate in bacteria?
Okay. Okay. You mean to say that whether any of the feeding
what is
events?
the effect of this circadian rhythm on the bacteria's development?
Yeah, so basically, this might be okay. They might be fine tuning their growth itself to these circadian rhythms. Okay, one is there, and second thing is that they might be adjusting their metabolic events to the circadian rhythms. I mean, there are various cellular events that are controlled by this bacteria. So, or even dispersal, proliferation, and all these things can be controlled by this circadian.
And what is the stimuli of this circadian rhythm? Like for
again, it's light. So light on and light off, and then.
Great. So since we are going, we have almost reached to end of this session, and we have a few questions in the chat box. A couple of actually 4 questions from Farheen. So I'll just club those and ask those questions to you. One is we talked about these sensors, like bacterial sensors. So can we use such sensors as target to prevent establishment of infection?
Yeah. So basically what Karin is asking is whether we can design drugs against these sensors and then prevent. Of course, definitely. And there are drugs which are actually being designed against some of the sensor kinases in pathogenic bacteria, which are these are early stages of development and early stages of research. But of course, these sensor kinases are being exploited as drug targets.
Right. So, and this, so another question which is asked by Farheen is, are there drugs that can target the biofilm? And I think there are studies which are.
There are drugs which can target the biofilm. Yeah, again, biofilm formation, and again, the signaling you just disrupt the signaling events that coordinate this biofilm. So for example, if it's a quorum sensing event which coordinates this biofilm, you can disrupt this quorum sensing events, confuse them, and then also you can dissociate this biofilm. Another interesting thing is that these are swimming bacteria, isn't it? So, and this bacteria, when they swim, they form a biofilm, they need to switch off their flagellar rotation, isn't it? So, and there are clutches in bacteria which has been described, bacterial clutch, which basically disengages this flagellum when they reside inside this biofilm. Okay, because if you end up producing flagellar rotation in a biofilm, then you're going to distort this biofilm, isn't it? So there are clutches and breaks in bacteria which just stops this flagellar rotation when they enter into this biofilm. And of course, you can either dissociate this biofilm by directly impinging on the matrix that holds this biofilm together, or I mean, of course, this matrix is produced by bacteria, isn't it? So then you end up interfering with the signaling mechanisms that produce this matrix, that ends up producing this matrix. And or what you can do is you can basically stop the communication between each of these bacteria, or interfere with this communication between each of these bacteria in this biofilm, which can end up distorting the biofilm and things like that. Of course, yeah, there are drugs which are targeted against biofilms. And it is very important that we use, and these drugs are also, in some studies, what they have used is these drugs are used in combinatorial therapy. So as I told you, so now there is no access for the antibiotic to the bugs which are residing at the center of this biofilm, isn't it? So now these drugs which goes inside, you might end up having a biofilm disrupting drug, which actually disrupts this biofilm, and then you use your antibiotic, which basically kills the kills the bacteria directly. Okay, those kinds of things can be used. And in fact, when I'm looking at this biofilm, there is another interesting aspect which happens in bacteria when they reside in this biofilm. So now let's say that it's just like antibiotic. This biofilm, the guys which are at the center of this biofilm, are not going to get enough nutrients, because only the ones which are at the periphery are going to get more nutrients, isn't it? And there is a fantastic study which is out there, wherein these guys which are at the center, they send signals out to this bacteria which are at the periphery, and tell them not to eat, so that the nutrients can basically dissipate or permeate into this center portion, and then these guys can feed. Okay, yeah, those kinds of activities also happen. Yeah.
Great. So amazing. So let's, I mean, I think we have covered almost most of the things about the bacterial cell cycle, and we got to know that bacteria, though exist as a unicellular organism, has a lot of complex world in that small space. And great. So thank you, Sunish, for joining us today and giving such an enlightenment.
Definitely, yeah.
This wonderful world of bacteria. And we thank our viewers to stay for being with us till this long, and being with us for like 2 seasons now, and 14 sessions. uh, and we are very, 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 very happy that you have been supporting us in our science outreach initiative. So as I promised in the beginning, that we have some news and announcement to make. So uh, in this outreach initiative, we will be talking to like audience or our viewers directly through Instagram live sessions. And I will not tell the dates. I'll not release the dates or from when we are doing it. So please follow us on Bugbears NCBS handle of Instagram, and stay tuned for updates. And you can definitely participate in our outreach initiative by sharing these videos.
Yeah, sorry, I just wanted to add something. So if anyone has any more questions, you can easily feel free to email me. I can just type my email out. Just hold on. Okay. So my email is, so is it visible?
Yes. So I'll just type in the chat box also.
Yeah. So you can definitely email me anytime if you have any more questions, or if you want to talk anything about bacteria, anything about bacteria. Yeah.
So that's nice of you. And so you heard it, you heard him. And so feel free to ask any questions about bacteria, bacterial cell cycle, differentiation, and anything that you can think of. And niche, and you can maybe type your questions in the comment box also. So feel free to share videos, and we will resume back these live sessions after a couple of months or so. And till then, stay safe. Please follow COVID appropriate behavior and get vaccinated if possible. Thank you. Till then, bye bye.
Bye.
Bye.